On to the nervous system. So we're going to do a quick review of the nervous system, and this will be included in your weekly review this week. And then we're going to talk about action potentials and some specificities along with that. As you can see here, we have our cell body of our neuron, which contains the nucleus. We have these projections called dendrites, and they are going to be the receiving end of our action potentials. And then the larger of these projections is called an axon. This opening part right here is called the axon hillock, but the rest of it is called the axon. And notice that this axon in particular is myelinated with these Schwann cells. This is a Schwann cell, so we know that this particular neuron is in the peripheral nervous system. Between these myelin sheaths, there are these open gaps of the axon, which are called the nodes of Ranvier. And this structure is important because it's going to impact how action potentials move through a neuron, depending on whether they're myelinated or not. So an action potential will come in through these dendrites, and it's going to cause changes within these cells, which we're going to talk about, and then propagate change throughout the axon and maybe even send an action potential through the axon.
This image is showing you different types of neurons. This top one is a multipolar neuron, which just means it has multiple projections coming off of it. And you can see that our dendrites collect or receive, and then the axon propagates the action potential down its length. This is a pseudo-unipolar neuron. The dendrites receive the action potential, and then again, it propagates down the axon. So one thing to notice that this is a one-way street. Things do not go back and forth, both ways. It's going one way in one direction only. So hopefully you remember a little bit of this from anatomy. This is showing you the neuronal pathways between the peripheral and central nervous system. So the central nervous system includes our brain and our spinal cord, which is shown here. The peripheral nervous system is going to include any nerves, and so it shows us the neurons or their axons or dendrites that are within those nerves. And then you can see that these dendrites are receiving an action potential. This is a sensory neuron, so this is going towards our central nervous system, so this would be considered afferent information. And then it can impact your central nervous system, which is going to be the integrating center. It's going to integrate or process that information somehow, and then it's going to cause an effect. If it is a somatic effect, as in like your skeletal muscle movement, notice it says skeletal muscle here, it is going to impact one motor neuron, action potential, and move one way. If it is an autonomic impact, it is going to impact a two motor neuron chain. So there's two motor neurons here. And we call this the presynaptic neuron and postsynaptic neuron, or preganglionic neuron, postganglionic neuron. And notice that it is impacting some visceral organs, smooth muscle, cardiac muscle, glands, because these are impacting your autonomic nervous system. And notice it is one way again. This is showing you the structure of a Schwann cell around an axon. You can see the nucleus here and how they roll around like a jelly roll. And again, Schwann cells are a neuroglial cell in your peripheral nervous system, whereas oligodendrocytes myelinate around the axons in the central nervous system. So, as we spoke about in the last video, we are interested in what happens with the membrane potential. And if you can remember, the resting membrane potential is anywhere from negative 60 to negative 80-ish millivolts, so usually somewhere around negative 70. And we're going to talk about these terms because we're getting into action potentials, and action potentials cause a change in our membrane potential. If it depolarizes, so notice this is at negative 70 here, if it depolarizes, that means it makes it less polar, and so it is going positive. It's going to make it more positive. The membrane potential will be above the resting membrane potential, which is around negative 70. If it repolarizes, that means it's going to go back down to being more polar, so more negative. So it's returning to normal, kind of like returning to negative 70. But if it hyperpolarizes, that means it's going past normal. And so it goes past or below our resting membrane potential. So we're going to use these words again. So we have our passive channels where ions can move through passive diffusion from high to low concentration. We have voltage-gated channels where the voltage, like whether it's positive or negative millivolt changes, is going to turn open or close these gates. And then we have chemically gated, which means if we have a receptor that binds to a specific ligand, then it may cause the gate to open. So membrane potential changes occur by ion flow through our membrane channels. And what is happening in a lot of these are these voltage-gated channels. These are going to open when we depolarize the cell. And remember, that means that it becomes less negative or more positive. And so what that does is it's going to open these channels that are usually closed in resting cells. And these are our sodium and potassium voltage-gated channels. So this is what it can look like. A voltage-gated channel is here shown closed. When our resting membrane potential goes upward and depolarizes, it is going to open up the channel so that ions can rush in or out, depending on which one we're talking about. But there is also a ball and chain here that can cause it to not close, but to inactivate. So this inactivation can occur during the refractory period, which we'll talk about in just a second. Okay, so we're about to get into an action potential. And we're talking about the wave of change in the membrane potential due to a movement of ions in the axon membrane. So there's going to be a change of the millivoltage in the membrane potential because of the movement of ions here. And this is occurring via diffusion. It's just ions that are going to go from high to low concentration because they can. And I want to remind you that this is capable because of that set up, really different concentration gradients set up by our sodium-potassium pump. In the steps of an action potential, and we can think about this at the neuron. I'm going to draw a neuron here. We have an action potential coming down our dendrites and then down our axon. What's going to happen is there are going to be ion changes that occur within the cell, which is going to change that millivoltage. So at rest, it is at negative 70 millivolts, but what happens is there's going to be so much change where it reaches a threshold, and once it reaches a threshold, it opens up these sodium channels. These are voltage gated. And if we reach our threshold, which is negative 55 millivolts, it is going to open up those voltage-gated channels. Remember that sodium is really high on the outside of our cell and not so much on the inside. So what's going to happen is it is going to rush into the cell. So sodium rushes in. Sodium is a positive ion. It takes our resting membrane potential and it increases it big time. And that's because those voltage-gated sodium channels have opened. So sodium crosses into the intracellular fluid and depolarizes the cell. This occurs until it reaches about positive 30 plus 30 millivolts. When it reaches plus 30 millivolts, the sodium channels are inactive. That whole ball and chain thing happens where they get inactivated. A ball gets stuck here, so they can no longer go through. It's not necessarily that they close, they get inactivated. So at this point, though, the voltage-gated channels of potassium have opened. So being at a plus 30 millivoltage causes the voltage-gated potassium channels to open. And now remember that potassium was really high inside of our cell. Those gates open, it's going to rush out because it's going from high to low concentration. As it rushes out, it's going to decrease, I don't know if you can see that color, decrease our millivoltage here caused by potassium moving out of the cell, and it even surpasses this negative 70 millivoltage. This is called repolarization. It's getting repolar. It's getting polar again. And then after it passes negative 70, this is called hyperpolarization because it's going even lower than our resting membrane potential. So it repolarizes the cell, and then it goes even past negative 70, all the way to negative 90. I want to remind you that negative 90 is potassium's equilibrium potential that we talked about in the previous video. So this is hyperpolarization. Once it reaches negative 90 millivolts, those voltage-gated channels can now close. And it stays this way until the sodium-potassium pump restores the concentrations and the potential goes back to negative 70 millivolts. Action potentials are an all or none event. They have to pass that threshold of negative 55 millivolts because that is what opens up that voltage-gated sodium channel. And if they pass through that negative 55 millivolt, there is going to be an action potential. As long as they go through this, there's going to be an action potential. A single stimulus is given to our neuron. It will activate or fire or give an action potential if the threshold is reached, if that negative 55 millivolts is reached. If it's not going to open those voltage-gated channels and it's not going to reach. And so this is what we mean by saying action potentials are either going to happen or they're not going to happen. And what that means is that you can't have like a big action potential and then a little action potential, right? Action potentials do not increase in size based off of the size of the stimulus. As long as there is a stimulus that can make it go above this threshold millivolt, there is going to be an action potential. What can change though, so it can't change its size of action potential, right? That's not a thing. We can't change the size of an action potential. What we can change is the frequency of action potentials. So here we have like a small stimulus, and notice there's action potentials occurring. Now we increase the stimulus and we have a lot of action potentials. So you don't change the size of your action potential, but you change the frequency or how many action potentials there are.